11 kilometers. That is the number. Not 11 kilometers of open water, 11 kilometers of descent, straight down, through increasing cold and increasing pressure and increasing darkness, until the ocean floor of the Challenger Deep receives you at a depth that no sunlight has ever touched and no surface weather has ever disturbed. The pressure at that depth is over 1,000 atmospheres, 1,000 times the weight of the air pressing down on your body right now. The temperature hovers just above freezing. The water column above weighs approximately 11 tons per square centimeter of surface it rests on. By every measurement that matters to biology, this environment should be sterile. The physical conditions at the bottom of the Mariana Trench exceed the tolerance limits of virtually every biological system that has ever been formally studied. And yet, when the first serious scientific expeditions began sending instruments to the Challenger Deep floor, what they found was not an empty abyss. Today, we're diving into what actually lives at the bottom of the deepest place on Earth, and why the biology discovered there has forced a fundamental revision of what science thought it understood about the limits of life. The Mariana Trench is not a single feature. It is a crescent-shaped scar in the floor of the western Pacific Ocean, stretching approximately 2,500 kilometers in length, formed at the convergence boundary where the Pacific tectonic plate subducts beneath the Mariana Plate. The Challenger Deep, the deepest surveyed point within it, sits at the southern end of this structure. The trench is geologically active. The subduction process that formed it continues today, generating seismic events along the boundary and creating a physical environment that is not merely static darkness, but an occasionally violent one, subject to pressure waves from seismic activity along the plate boundary below. The floor of the Challenger Deep receives material from the entire water column above it, sinking organic particles, dead organisms, chemical precipitates, in a slow, continuous rain of biological and chemical debris that accumulates over geological time. This material, called marine snow, is the energy source for everything that lives there. Not sunlight, not photosynthesis, chemosynthesis, decomposition, and the slow digestion of what falls from above. The hadal zone, the collective term for ocean environments below 6,000 meters, is powered by death falling from above, and what it does with that energy is one of the most remarkable stories in the history of biology. Before anything else lived in the Mariana Trench, the water column itself was colonized. Between the surface and the floor, pressure increases in a continuous gradient, and the organisms that inhabit different depth zones within the trench water column represent successive communities of specialists, each calibrated to the specific pressure and temperature range of their preferred depth. Midwater jellyfish, xenophyophores, and deep-sea fish occupy the upper portions of the trench water column, animals that are extraordinary in their own right, but still operating within the general parameters of deep-sea biology as understood from other oceanic environments. As depth increases past 4,000 meters, then 5,000, then 6,000, the community composition changes in ways that become increasingly difficult to predict from shallower water equivalents. The animals below 6,000 meters, the true hadal zone, are not simply deeper versions of what lives above them. They are a distinct ecological community with its own specialists, its own food web architecture, and its own biological innovations that appear nowhere else in the ocean. The transition across the 6,000 meter boundary is not a gradient, it is a threshold. Foraminifera were not supposed to be there. These single-celled organisms, amoeba-like protists that build intricate shells of calcium carbonate or agglutinated sediment particles, had been studied extensively in shallower ocean environments where their biology was considered reasonably well understood. Then samples from the Challenger deep floor revealed giant foraminifera, xenophyophores, reaching sizes of up to 20 centimeters, making them among the largest single-celled organisms ever documented on Earth. Syringamina fragilissima and related species form irregular branching structures from sediment particles cemented together, sprawling across the deep sediment surface in forms that look more like coral fragments than single cells. The size alone defied existing models of single-cell biology. The relationship between cell size, metabolic rate, and the diffusion limits of oxygen and nutrients was thought to impose strict upper bounds on how large a single cell could grow. The xenophyophores at hadal depths ignored these bounds entirely. Their metabolic strategy, their reproductive biology, their precise relationship with the chemical environment of the deep sediment, these remain incompletely understood. What is certain is that they are there, they are large, and nothing in the shallower ocean prepared science for them. The xenophyophores are not passive structures. They actively accumulate heavy metals, barium, lead, uranium, from the surrounding sediment at concentrations far exceeding ambient levels, incorporating these elements into their cellular mass in quantities that would be toxic to virtually any other organism. Why they do this is not fully established. Some researchers propose that the heavy metal accumulation is a byproduct of their sediment processing feeding strategy. Others have suggested it may serve a structural or metabolic function that has no equivalent in any studied biology. What the xenophyophores do unambiguously is create habitat. Their branching structures provide physical complexity to an otherwise flat sediment surface, and the microenvironments they create, their interiors, their surfaces, the sediment they disturb, host communities of smaller organisms that would not be present on bare sediment. A single large xenophyophore is a small ecosystem. A field of them, which is what the Challenger deep floor contains in certain areas, is an ecological infrastructure that supports biological diversity at the deepest point on Earth in ways that have only recently begun to be mapped. 8 kilometers down, something is swimming. Snailfish, members of the family Liparidae, have been filmed and collected at depths that would have been considered impossible habitat for vertebrate life as recently as 3 decades ago. In 2023, an individual snailfish was filmed at 8,336 meters in the Izu-Ogasawara Trench, the deepest vertebrate ever recorded. These are not large animals. They are pale, translucent, gelatinous-looking fish, their bodies soft, and their skeletons incompletely mineralized, a structural adaptation to pressure that removes the rigid calcium architecture that would otherwise be crushed. But the biology of their survival at these depths requires more than structural softening. The cell membranes of all living organisms are composed of phospholipid bilayers that become rigid and dysfunctional under high pressure. At hadal depths, the pressure would stiffen these membranes to the point where ion channels cannot open and cellular signaling collapses entirely. Hadal fish compensate with elevated concentrations of trimethylamine oxide, TMAO, a small organic molecule that counteracts pressure-induced membrane stiffening at the molecular level. The TMAO concentration in hadal snailfish tissue increases linearly with depth, as if the animal's biochemistry is actively titrating its own pressure compensation. At the deepest depths where snailfish have been found, TMAO concentrations approach a theoretical upper limit, a ceiling imposed by the osmotic consequences of accumulating too much of the molecule. This ceiling may define the absolute maximum depth at which any vertebrate can physiologically function. If it does, snailfish are already at that ceiling. They have reached the bottom of what vertebrate biology can tolerate, and they are thriving there. The snailfish do not simply survive at these depths, they reproduce there. Juvenile snailfish have been recovered from hadal trap samples, indicating that the full life cycle, from birth through growth to maturity, occurs within the extreme pressure environment of the trench. This is biologically significant in ways that extend beyond the pressure tolerance of adult tissue. Development, the process by which a single fertilized cell divides and differentiates into a complex multicellular organism, is exquisitely sensitive to environmental perturbation. The biochemical signaling cascades that guide embryonic development were not designed with extreme hydrostatic pressure in mind, and yet snailfish embryos develop normally at hadal depths, meaning that every pressure-sensitive step in the developmental program has been modified to function under conditions that would abort development in any shallow water fish. The snailfish is not merely a pressure-tolerant adult, it is a pressure-tolerant organism at every stage of its biology, from the first cell division onward. The sediment itself is alive in ways that are difficult to overstate. 1 teaspoon of deep ocean sediment from hadal depths contains millions of microbial cells, bacteria and archaea that live in a state of extraordinarily slow metabolism, dividing perhaps once every 1,000 years, subsisting on the chemical energy of ancient organic material buried in the sediment column. These are not organisms operating at the edge of survival. They are organisms that have redefined the concept of survival itself, existing on energy budgets so small that the entire framework of metabolic rate that applies to surface biology becomes essentially meaningless when applied to them. Some of the microbial cells recovered from deep ocean sediment cores appear, based on radiocarbon dating of the surrounding sediment, to have been alive and metabolizing for over 100 million years, not as dormant spores, but as continuously active cells running at metabolic rates so slow they are nearly impossible to measure with conventional instruments. The boundary between living and non-living becomes philosophically ambiguous at these timescales and energy levels. What does it mean to be alive if your metabolic rate is so low that a single cell division takes a millennium? The answer the deep sediment community provides is that life does not require speed. It requires only continuity, the unbroken thread of chemical reactions that separates biology from geology. These microbial communities are not uniform. The sediment column of the Challenger Deep contains distinct microbial communities at different depths, each community reflecting the chemistry and organic content of the sediment layer it occupies. Surface sediment communities process recently deposited organic material. Deeper sediment communities metabolize progressively older and more chemically recalcitrant organic compounds. At the very base of the sediment column, where the sediment meets the basaltic rock of the ocean floor, communities exist that appear to be deriving energy not from organic material at all, but from chemical reactions between the rock and the water, a form of chemolithotrophy that was discovered in deep ocean settings and has since been found to extend throughout the oceanic crust. The rock beneath the Mariana Trench is not dead. It is inhabited. The biosphere extends downward through the sediment and into the crust itself, and the total biomass of this subsurface microbial world may be comparable to or exceed the total biomass of all life visible at the surface of the planet. Amphipods arrived at the Challenger deep floor before any human instrument did. These crustaceans, small shrimp-like scavengers belonging to several hadal specialist genera, most notably Hirondalia gigas, occur at the deepest points of the trench in densities that surprised every expedition that measured them. They are not rare. They aggregate in large numbers around any organic material that settles on the floor, including the bait used in scientific traps, which they can strip to bone within hours of deployment. Their body plan is relatively conventional for a crustacean, but the biochemistry underlying their pressure tolerance is not. Hirondalia gigas produces unusual lipid compositions in its cell membranes that maintain fluidity at extreme pressure. It also produces digestive enzymes, cellulases, that allow it to digest wood cellulose, extracting energy from sunken wood debris that falls to the trench floor from terrestrial sources carried to sea by rivers. An amphipod at 11 kilometers depth, digesting wood that started its journey in a forest on a Pacific island, processing it with enzymes that have no close equivalent in any shallow water crustacean. This is the hadal ecosystem operating as an integrated system that connects the deep ocean. Floor to the surface world through pathways that are still being traced. The population genetics of hadal amphipods have added an unexpected dimension to the understanding of deep trench ecology. Studies comparing amphipod populations from different trenches, the Mariana, the Kermadec, the Philippine, have found surprisingly high levels of genetic exchange between populations separated by thousands of kilometers of open ocean floor. The mechanism of this dispersal is not fully established. The conventional assumption that individual trenches represent isolated evolutionary islands has been challenged by genetic data showing that amphipod larvae or adults can apparently cross the deep ocean floor between trench systems, maintaining gene flow across distances that surface intuition would suggest are insurmountable barriers. The deep ocean floor, which appears from above as a featureless barrier between discrete habitats, may function for some hadal organisms as a connective matrix rather than an obstacle. The geography of deep sea evolution does not obey the rules inferred from surface ecology. Polychaetae worms occupy every available niche in the hadal sediment. These annelid worms, relatives of the earthworm, adapted to marine environments, are the most species-diverse group of macrofauna in most hadal sediment samples. They process sediment, prey on microbes and smaller invertebrates, and are themselves prey for larger animals. Some hadal polychaetes are predatory, equipped with complex jaw structures capable of capturing other macrofauna. Others are deposit feeders, processing bulk sediment for its microbial and organic content. The functional diversity of the polychaetae community in a single hadal sediment core sample can rival that of shallow water benthic communities with vastly greater energy input, a finding that challenges the intuitive assumption that extreme energy limitation should produce simplified, low diversity ecosystems. The hadal sediment community is not simple. It is complex, functionally diverse, and organized in ways that reflect millions of years of ecological refinement in an environment that surface biology predicted would support almost nothing. Holothurians, sea cucumbers, move across the hadal sediment surface in numbers that have astonished every expedition that has imaged the Challenger deep floor. In some areas of the trench floor, holothurian densities are high enough that they appear in nearly every frame of seafloor footage. These animals process enormous volumes of sediment, ingesting it for its organic content and excreting the processed material behind them in trails visible on the sediment surface. The holothurian trails photographed on the Challenger deep floor represent, in aggregate, a significant processing of the entire sediment surface, a biological reworking of the deep trench floor that redistributes organic material, mixes sediment layers, and creates the physical and chemical heterogeneity that other species depend on. The sea cucumber, one of the most morphologically simple of all echinoderms, is the dominant large animal on the floor of the deepest place on Earth. It is doing, at 11 kilometers depth, what it does in shallow coastal waters, eating sediment and moving slowly. The simplicity of the behavior belies the biological sophistication required to perform it at this depth. Not everything in the Mariana Trench fits neatly into established taxonomic categories. Repeated sampling expeditions have returned organisms that defy confident classification, specimens that share characteristics of multiple known groups without belonging clearly to any of them, or that represent lineages with no close known relatives anywhere in the modern ocean. The isolation of hadal environments, the extreme depth, the physical barriers between individual trenches, the long geological history of the subduction zones that created them has produced evolutionary divergence on timescales and at rates that shallow water biology does not prepare. Researchers to expect. Some hadal species appear to have been isolated in their trench environments for tens of millions of years, accumulating genetic differences from their closest surface relatives at rates that reflect the slow generation times and low metabolic rates of deep sea life. The hadal zone is not a uniform global environment. Each major trench is, in an important sense, its own evolutionary island, with its own community of specialists shaped by the specific chemistry, geology, and history of that particular depression in the ocean floor. The acoustic environment of the Mariana Trench is one that science has only recently begun to characterize. The deep ocean is not silent. It is filled with sound, biological sounds from animals communicating, feeding, and moving. Geological sounds from the active subduction zone that created the trench. Anthropogenic sounds from surface shipping, sonar, and industrial activity transmitted downward through the water column. The Challenger Deep acts as an acoustic collector. Its geometry focuses and amplifies sounds from a wide surrounding area, concentrating them at the floor. A hydrophone deployed at hadal depths in the Mariana records not just the local acoustic environment, but sounds originating thousands of kilometers away, from shipping lanes and distant storms and seismic events across the Pacific Basin. What this acoustic environment means for the biology of the trench, whether hadal animals use sound for communication or prey detection, whether the elevated noise levels from anthropogenic sources affect behavior or physiology in ways that are measurable, is a research question that is only beginning to be addressed. Plastic arrived before the cameras did. When the first modern high-resolution imaging expeditions reached the Challenger deep floor, they found, alongside the xenophyophores and amphipods and pale bacterial mats, human-produced plastic debris, plastic bags, candy wrappers, fragments of fishing line, material manufactured within the last century, already present at the deepest point on Earth's surface, incorporated into the sediment that also contains microbial cells that may be 100 million years old. The contamination of the Challenger Deep with surface-produced synthetic material is not a marginal finding. A 2019 analysis found microplastic particles in every amphipod specimen sampled from 6 deep ocean trenches, including the Mariana. The animals at the bottom of the world's deepest trench are eating human plastic. The ecosystem that evolved over millions of years in complete isolation from the surface world has within a century of industrial production been reached by its chemical byproducts. The full biological consequences of this contamination, how microplastics interact with the membrane biochemistry of hadal organisms, how they affect the microbial communities in the sediment, what happens to the food web when every organism in it is ingesting synthetic polymer particles, are unknown. The experiment is running, the results are not yet in. The chemistry of the hadal water column itself is unusual in ways that are still being characterized. Deep trench water is not simply a colder, more compressed version of the water above it. The isolation of the trench water from the broader ocean circulation creates distinct chemical signatures, elevated concentrations of certain dissolved gases, anomalous dissolved organic carbon profiles, unusual pH gradients near the sediment surface. The chemistry of the water interacts with the chemistry of the sediment and the chemistry of the organisms living in both, creating a set of environmental conditions that are specific to individual trenches and that change with depth within a single trench. Organisms living at the floor of the Challenger Deep are not simply dealing with high pressure and cold temperature. They are dealing with a specific chemical environment that is found nowhere else on Earth that varies from the floor of the Mariana to the floor of the Tonga Trench to the floor of the Puerto Rico Trench, and whose biological consequences have barely begun to be systematically studied. What the Mariana Trench ultimately represents is not merely a geographic extreme. It is a boundary condition, a place where the physical parameters of the environment push against the limits of what biology can endure, and where life, having reached those limits, has responded not by retreating, but by innovating. The Tamao system in hadal fish, the membrane lipid chemistry of hadal amphipods, the pressure-tolerant enzyme systems of hadal bacteria, the extraordinary size of hadal xenophyophores, the developmental pressure tolerance of snailfish embryos, the cellulase-equipped digestive systems of wood-eating amphipods. Each adaptation is a specific biological answer to a specific physical question posed by the environment. The questions at 11 kilometers are more extreme than anywhere else on Earth's surface. The answers evolution has produced there are correspondingly more extreme than anything found in shallower water. And the most unsettling implication of what science has found in the Challenger Deep is not that life exists there, it is that life appears to be thriving there, in ways and by mechanisms that the biology of the surface world did not predict and still does not fully explain. Every expedition that has reached the hadal zone has returned with organisms that were not previously known to science. Every sediment core pulled from the deep trench floor contains microbial communities whose metabolic strategies are still being characterized. Every trap deployed at maximum depth recovers amphipods whose biochemistry contains surprises. The floor of the Mariana Trench has been directly visited by human instruments fewer times than the surface of the moon has been walked on. The proportion of its biology that has been formally studied is a fraction of what almost certainly exists there. New imaging technologies, autonomous underwater vehicles capable of operating at full ocean depth, new generation lander systems with higher resolution cameras and more sensitive chemical sensors are reaching the hadal zone with increasing frequency, and each new expedition returns data that revises the previous picture in ways that were not anticipated. The trench is not a finished subject, it is an active frontier, and the rate of discovery there is accelerating rather than slowing as technology improves. The implications of what lives in the Mariana Trench extend well beyond oceanography. The discovery of active biology at extreme pressure, temperature, and energy limitation has fundamentally changed the assessment of where life might exist elsewhere in the solar system. The subsurface oceans of Europa, Enceladus, and other icy moons of the outer solar system are environments that share key characteristics with the hadal zone, extreme pressure, cold temperature, absence of sunlight, potential chemical energy sources from water-rock interactions. Before hadal biology was characterized, these environments were considered marginal candidates for life. After it, they are considered serious ones. The organisms of the Mariana Trench have not only expanded the definition of habitable environment on Earth, they have expanded it across the solar system. What lives at the bottom of the deepest trench on this planet has become a reference point for what might live in the depths of oceans on other worlds. The floor of the Mariana Trench has been at the bottom of the Pacific Ocean for tens of millions of years. The subduction zone that created it has been active since before the first grasses appeared on land. The microbial communities in its sediment have been metabolizing slowly across timescales that make all of recorded human history a thin recent mark at the very top of the sediment column. The snailfish swimming in its darkness have been evolving there, generation by slow generation, since before the ancestors of modern humans stood upright. And the darkness itself, the absolute, total pressurized darkness of the Challenger deep floor, has been waiting, patient and complete, for science to arrive with enough technology to begin to understand what it contains. What science has found is extraordinary. What it has not yet found, in the kilometers of unexplored sediment and the vast unmapped floor of the deepest place on Earth, may be more extraordinary still. The trench does not give up its contents easily. 11 kilometers is a long way down, and everything living there has had a very long time to become exactly what the darkness required.